Hello everyone, welcome back to another video of Biotech. This is the series of genetics subject and this is the second video in which we will talk about the genes, and this video has two parts. In the first part we will discuss the introduction of genes. We will also discuss the historical background and the discovery of Mendelian work, and we will also look at the central dogma of our molecular biology, that how DNA is transcribed into RNA and then how RNA is translated into a protein molecule, which is the building block element of life. Now secondly, we will also discuss the molecular structure of gene, how what DNA contain, what is the double helix structure, what is DNA, and what is the genetic code. We will also discuss the gene function, for example translation, transcription, the expression of genes, and the function of protein in this video. And we will also cover the epigenetic mechanism, which comes in the category of gene regulation. This topic will explain you that how a gene is expressed into any other sequence, and this mechanism, it is the epigenetic mechanism. So these things will be discussed and the topic of today's video. Let's start this video by looking at the introduction of genes. So genes are the segment of DNA, and that segment contain the instruction for building and maintaining a living organism, and these information, they are passed from our parents to offspring, and this is the basis of heredity, which is the genetic material. Now let's understand this topic by looking at the introduction to the genes. So basically in biology, genes have two definition. The first definition is given us by Gregor Mendel, and the second definition is about the molecular genetic mechanism. So in the first definition, what Mendel tell us, so Mendelian genomic data says that gene is the basic unit of heredity material. Mendel said that gene is the only unit that contain our genetic information, those information that we obtain from our parents. And in the second definition, the molecular gene tells us that it is the sequence of nucleotide, gene is the sequence of nucleotide in DNA, which is present in DNA molecule, and that molecule is transcribed into protein. But how it is transcribed into protein, that molecule is further first transcribed into RNA, and then RNA it is translated into protein. Now there are two types of that molecular genes. How it has two types? So the first type, it is called coding genes, and the second, it is called non-coding gene. So we can also say that it is a protein coding genes and a protein non-coding genes. Those section in this diagram, you can see that the coding region, it is termed as exon, and the non-coding region, it is called intron. And you can also see in this diagram that how exon, it is referring to the transcription mechanism, because the non-coding gene are not involved in the expression pattern. Only the coding, protein coding gene are involved in this mechanism to form a protein molecule. So how, what is the promoter? At the promoter region, a DNA is attached, and then the exon region, the intron region, it is doing the transcription mechanism and a formation of RNA molecule. And then RNA molecule, it is spliced to form messenger RNA, and that messenger RNA, it is translated by the process of translation to form an amino acid chain. And now the production of amino acid chain, it tells us that the formation of protein is occurring, because amino acid is the building block of protein. How different amino acid is combined and it form a protein molecule. Now we can see that historical background and the history of Gregor Mendel work, they tells that the pea plant is the basis of the fundamental laws of inheritance, because he worked on the pea plant. And what he did, he deduced that genes come in pairs. No gene are present in a single form. Every gene is present in a pair form, and that genes are inherited as distinct characters and distinct units from one parent to other parent. For example, one character you obtain from your father, and the other character you may obtain your mother, are both of them are involved in this factor. So Mendel tracked the segregation of parental genes and their appearance in the offspring as a dominant and recessive traits. Dominant traits are those traits that are strongly highlighted on an organism. For example, if your father has a blue eye, if the child have a blue eye, that character in child is dominant from the father the character. Now if the trait, what happens in the recessive trait, the trait is present but it is not very highlighted, that trait is termed as a recessive trait. Dominant are those traits that has that overwhelm the character. Now we will see the central dogma of molecular biology, what we call why it is central dogma. So the conversion of DNA to RNA and then the conversion of RNA to form a protein molecule, this is the definition of central dogma of molecular biology. So what central dogma of molecular biology is that a gene that is present in DNA, it is transcribed into an RNA by the process of transcription, and that RNA, it is translated into protein by the process of translation. So you got my point. Now that RNA, it is also converted into that DNA molecule by the process of reverse transcription. The molecular structure of genes, it tells us that the genes are present in DNA structure, and that DNA is a double helix structure. So we need to dive into the structure of DNA. You can see on the screen the structure of DNA. Now, the structure of DNA molecule that carries our genetic information, and that DNA is made up of two strands. That two strands are coiled around each other to form a double helix. So, double helix structure, single one to form a coiled double helix structure. Each strand is composed of a series of nucleotides, and that nucleotides include adenine, which is represented by capital A, thymine, which is represented as capital T, cytosine as capital C, and guanine as capital G. Now the molecular structure of gene, it tells us that genes that is present in DNA, and that DNA is a double helix structure. That structure was given us by Watson and Crick, who proposed the structure of DNA in 1950s. The other thing that we see in the structure of genes is that the DNA contains nucleotide on its both strands, and that nucleotides contain that bases which is adenine, thymine, cytosine, and guanine. Now that DNA, it forms a chromosomal structure. The organization of DNA into chromosome. DNA are organized into different strands, and that strands, it forms the chromosome structure. So difference between genes, exon and introns we will cover in the upcoming videos, as well as we will also cover the genetic code. For example, the codon, the codons, the non-codons regions, the coding regions, the non-coding regions, and their role in the synthesis of protein. We will cover that in the topic of protein synthesis, their formation and their structure. So you will be clear about that topic in the next video. Now let's look at the gene function. What is gene function? So gene function are being transcribed into RNA, which is then translated into proteins, and that proteins are the workhorses of life, which is also known as the building block and the structure element, because protein gives you structural support. It performs a variety of functions that are necessary for living a good life, and the process starts with transcription, where the DNA sequence of the gene is copied into mRNA, and that messenger RNA, it is translated to form RNA by the process of splicing, and then it converted into a protein molecule, which is the molecule that gives strength to your body, as well as it supports your body. So the expression of genes tells us that how gene turn off and turn on. For example, when it is required to form to form a protein molecule, the gene turn on and it express. But when it is not required to form a protein molecule, the gene molecule turn itself off by the help of promoter and enhancer region. And when the protein form, it performs their different function. As I mentioned, that we will cover the topic of protein, its structure and function in the next video, so you have to be clear about this topic. But we will just look at the overview of this protein in this video, and the next part of this video we will discuss that in that. So the structural protein, we have enzymes, which are also a protein in nature, and that enzymes help us to speed up the chemical reaction in the body, as well as the signaling molecule that signals our body in case of any harm or in case of anything. For example, you may be injured, and that signaling molecule move towards that part of that injury and help you cure that, to stop the bleeding or to cover the process. So we have also discussed that intron is called the non-coding region of the gene, and exon are the coding part of the gene. Now let's look at the gene regulation process. So gene regulation is a complex and a finely turned process, and this process ensures that genes are expressed at a right time, in a right cell, in a right place, and at the right amount of time. So how gene is regulated? An epigenetic mechanism like DNA methylation and histone modification, that processes occurs which plays a crucial role in regulating the gene expression without altering the sequence of DNA. So it says that the sequence of DNA, it is not changed in the process of DNA methylation and DNA and histone modification, and gene is regulated in to form a protein. So both DNA methylation and histone modification processes are involved in establishing a pattern of gene repression and the development of protein. So but certain form of histone methylation, it causes a local formation of heterochromatin structure, and which is readily reversible process, and DNA methylation, it leads to the stable long term repression. So what happened in the DNA methylation process, a methyl group is attached to the promoter region of DNA, and that methyl region attachment to the promoter region of DNA, it is called DNA methylation, and you can see this in the image. And what happened in the histone modification, an acetyl group, which is an acetyl group is attached to the DNA molecule to perform the specific function. So in the DNA molecule, a methyl group is added and it is called DNA methylation, and in the histone modification, a methyl group is attached, that is called, it is called histone modification. So histone modification involves the post translation modification. For example, after the translation, for example, when protein is formed, the process of histone modification occurs, so it is called as post translational modification of specific amino acids. But that amino acid influenced the overall structure of histone protein, because histone is a protein, and the amino acid, it changed the structure of that protein, and changes in that protein structure may influence the function of that protein, and also it also results in the incomplete DNA unwinding, which is also the effect of the transcription activity. Now what happened in the non-coding and non-coding RNase region, and what do they do? So the non-coding RNA region, small interfering RNA like siRNAs and microRNAs like miRNAs, they act as an RNA interferences, and this is the pathway to silence the gene expression. For example, if there is no requirement of gene expression and there is no requirement of protein formation, these microRNA and small interfering RNA, they involve in that process, and what they do, they do the transcriptional and post transcriptional level modification. The next we will cover to the second part of this video. Thanks for watching this video up till now, and we will complete this video on the topic of genes in our next lecture.